Assalamu alaikum. So in this part, we'll be talking about intermediate filaments, the third cytoskeletal structure that exists in cells. So we first talked about the actin filaments, then we talked about the microtubules, and here we'll be talking about the intermediate filaments. So what are these filaments? Well, they have a diameter that is intermediate between actin filaments and microtubules. So microtubules are the thickest of the three. Actin filaments are the thinnest, and intermediate filaments are in between. So what they do is that they provide mechanical strength to cells and tissues. So this is the real skeleton of the cell if we want to compare them to actin filaments and microtubules. They provide a scaffold, a structure, a three-dimensional structure, again, cytoskeleton. And what it does is that it integrates the different components of the cytoskeleton. That is, what these do is that they connect the microtubules to the actin filaments. And they also organize internal structure of the cell. That is particularly the nucleus. So they maintain the overall structure of the nucleus. They maintain the overall structure of the cell. Of course, we talked about the actin cytoskeleton, we talked about the microtubules, and we said that they support the cell structure and cell shape, except that they are dynamic, meaning that the cell shape changes with external factors, stimulants, molecules that stimulate or induce cells to move or to secrete certain products. These intermediate filaments are less dynamic. They are a bit more rigid in the way they connect to each other. There are 5 different types of intermediate filaments, and each one of them either has its own chemical characteristic or functional characteristic and tissue or cell distribution, unique cell distribution. So we have types 1 and 2. Okay, so we have 5 types. Types 1 and 2 are found in epithelial cells, and they synthesize, the intermediate filaments are synthesized as acidic filaments or neutral or basic filaments. So type 1 intermediate filaments are acidic, type 2 are more neutral or basic. A good example is keratins, and keratins exist in epithelial cells. Now these keratins, we covered them in the biochemistry course, and we said that these keratins are important structural proteins, and they exist in hair or nails. And we said that they become harder if there are more disulfide bonds existing within the intermediate filaments. So there are more disulfide bonds in the keratins in nails versus hair. Now, soft keratins also exist in the cytoplasm of epithelial cells. Okay, so they can exist as hard keratins in nails, somewhat hard in hair, and soft keratins also exist inside cells. Now, type 3 include vimentin, a special type of intermediate filaments that exist in fibroblasts, smooth muscle cells, and white blood cells. There's also a type 3 intermediate filament known as desmin, and this specifically exists in muscle cells. We'll be talking about this intermediate filament. We have type 4 intermediate filaments known as neurofilaments, and they exist in the axons of neurons, nerve cells. And we have type 5 type 5 intermediate filaments are the nuclear lamins. We talked about them before, like lamin A, for example. They maintain the shape and structure of the nucleus. So again, these are the different types. So we have type 1, acidic keratins, type 2, basic keratins, and they exist in epithelial cells. Type 3, intermediate filaments include desmin, so we talked about this, and vimentin. Vimentin exists in mesenchymal cells. Okay, desmin exists in muscle cells. We talked about type 4 briefly. We'll talk about it in more details in the following slides. And these include neurofilaments in nerve cells. Type 4, I'm sorry. Type 5 are the lamins, like lamin A and lamin B. They maintain the nuclear structure. They are the nuclear skeleton. So the way they look, or they are composed of a polypeptide, the basic structure is a polypeptide chain that is helical, and it has a rod domain. Okay, so there is a rod domain that exists in the center. In the ends of the polypeptide, we have a three-dimensional structure. We have a domain at the N-terminus and the C-terminus. And these domains are more globular in structure. And these are the domains that determine the specific function of the intermediate filaments. So let's talk about how this polypeptide forms the intermediate filaments. So basically, we have this single polypeptide. We have the formation of a dimer. So they wrap around each other. They wind around each other, um, uh, forming a coil-coil structure. So it's like a coil. Okay, so it's like it, it keeps on rotating, basically, having the structure right here. Then we have these dimers, two dimers associate with each other, forming a tetramer. So you have this interaction. They look anti-parallel. So you have the N-terminus and right on the interacting sort of with the C-terminus of another dimer. So they are anti-parallel in structure, but they're not parallel exactly on top of each other. Rather, they are staggered, meaning they are a bit sliding off each other a little bit. Then we have these tetramers forming associate end to end, forming what is known as a protofilament. And we have a number of these protofilaments coming together. We have 8 of them coming together, forming a larger structure known as a filament. That's the intermediate filament. So it's rope-like. So you have all of these helical structures coming together, forming a thick filament. So that's the basic structure of the intermediate filaments. Notice that there is no polarity like the actin cytoskeleton and the microtubules. Both of these other structures have a plus end and a minus end, an end that keeps on growing and an end that keeps on shrinking or depolymerizing or dissociating. It's getting shorter and shorter, and another end, a plus end, that keeps longer and longer, and they are regulated. So the intermediate filaments are actually more stable than the actin filaments and the microtubules. They are not as dynamic. They're not changing. They're not regulated by GTP. Rather, they're regulated by phosphorylation, like lamins and vimentins. And this phosphorylation results in the dissociation of the filaments. Now, note that this phosphorylation occurs at the globular head groups or the tail groups that exist at the ends, at both ends of the filaments. So this phosphorylation results in the dissociation of the filaments and the protofilaments as well. So where do they exist? Well, they exist all over the cell. Uh, Particularly, there is a concentration of these intermediate filaments around the nucleus. So what they do really is that they support the nucleus inside the cell. And we talked about the importance of these intermediate filaments in a sense that if they are mutated, they can affect gene expression, the activity of genes, and resulting in abnormal gene activity, resulting in a number of diseases like Mary-Charcot tooth disease. Now, in muscle cells, I just want to mention this intermediate filament known as desmin. We said that desmin, which is type 3 intermediate filament, exists in muscle cells. And what it does is that it connects the actin filaments to each other. So they play a major role in organizing the actin filaments inside muscle cells, and as a result, they sort of control or regulate muscle contraction. So they do play an important role in muscle cells. Now, in neurons, intermediate filaments are very important. And what they do is that they connect the microtubules to the actin filaments. So you have this intermediate filament, okay, like plectin, for example, in neurons, and they support the microtubules and the actin cytoskeleton. So if we look here, for example, we notice that the actin filaments exist nearby the plasma membrane of nerve cells. Microtubules are these thick green filaments in the axons. So this is an overview of the axon. The intermediate filaments, the plectin, exist right in the middle, connecting the microtubules to the actin cytoskeleton, to the actin filaments. So what they do is that they really support the axons, um, providing, again, mechanical stability of the cell and overall, and the axon specifically, and overall the whole cell. Well, let's talk about um, intermediate filaments in epithelial cells. Now, there are specialized structures in epithelial cells. We talked about, for example, we talked about uh, um, focal adhesions. We talked about adherens junctions. We talked about desmosomes as well previously. Well, we have these two specialized structures at the plasma membrane of epithelial cells. And these structures support cell-cell interaction, cell matrix interaction. They support the whole tissue. And these structures are known as desmosomes and hemidesmosomes. Desmosomes exist at cell-cell contacts. Hemidesmosomes exist at the cell matrix uh, uh, points of interaction, cell substratum contacts, the cell and the surface that they interact with at the bottom of the cell. Now, desmosomes, what we have in here is that we have this interaction between uh, receptors, transmembrane proteins of both cells. So one coming out of one cell and the other comes out of the neighboring cell. And they contact each other. An example is cadherin-like transmembrane proteins. And they interact with intracellular proteins, and these intracellular proteins interact with intermediate filaments. Okay, so it's really similar to adherens junctions, where we have the cadherins interacting with the actin cytoskeleton via catenins. Here we have the interactions between the receptors and the intermediate filaments, also via other intermediate filament binding proteins. Now, hemidesmosomes, on the other hand, we have this interaction between transmembrane receptors on the cell surface, like integrins, and we talked about integrins before, and these interact with the intermediate filaments inside the cell via specialized proteins as well. So this is similar to, and these integrins, by the way, interact with the matrix proteins. So we have this interaction, this connection, indirect connection between the intermediate filaments and the matrix proteins outside. So anything that happens outside the cell can be sensed inside the cell by changing the structure of the intermediate filaments. So this is similar to the focal adhesions, the specialized structure of the actin filaments that we talked about, and how actin filaments are connected to the extracellular matrix. Just to show you examples of the importance of intermediate filaments. In one famous experiment, one of the intermediate filaments was mutated in mice, and that resulted in the existence, the development of blisters, of injuries to the skin. The skin became quite vulnerable and susceptible to injury. It became mechanically sensitive to pressure, stress, abrasions, and so on. blisters يعني حبوب، بثور. abrasions الكلمة اللي حكيتها يعني احتكاك أو خدش. The same thing happens in humans, by the way. There's a condition known as the epidermolysis bullosa simplex, which is caused by a defective keratin mutations in the keratin gene, and that results again in defective skin that is susceptible, that is vulnerable, that becomes injured quite easily because it's not stable, it's not strong. We talked about ALS, we talked about amyotrophic lateral sclerosis, ضمور العضلات, that results in abnormal muscles. This disease is, we talked about before, mutations in lamin A, the intermediate filament nuclear skeleton protein. We talked about it in the microtubules lecture, and we said that abnormalities in the keratin, abnormalities in the microtubule motor protein kinesin can cause ALS. It can also be caused by defective neurofilaments, the intermediate filaments that exist in nerve cells. Okay, and the most famous person affected by ALS is Stephen Hopkins, the writer of this memorable book, The Brief History of Time. So, this figure is taken from this article right here from 2022, and it talks about ALS and the significance of the cytoskeletal protein, this cytoskeleton, in in ALS. Defective keratin, defective um, actin binding proteins, defective microtubule motor proteins can all cause ALS. And that shows how these cytoskeletal systems are connected to each other and how the three of them are important in in the biology of nerve cells, in the normal physiology of the central nervous system. This table shows the differences and similarities between the three cytoskeletal proteins in terms of structure, in terms of thickness, in terms of the proteins that make up the cytoskeletal structures and the functions as well. Allah